Thank you so much. Currently, I'm actually the director of preclinical AI at scientist.com, but thank you again for the introduction, and thanks to the organizers. I do hope that by the end of this talk, at least all of you here and those listening will find this to be an invaluable resource whenever you're looking for tumor models to test your drug candidates, or potentially when you're looking to outsource any work. All right. So that being said, I'm really excited to talk about our tumor model resource, again, which we call the Disease Model Finder at scientist.com. And I'm going to be going through all the ins and outs of that database, talking about some stats, the kind of models and distributions that we have in our database. But I'm also very excited to talk about how we are able to obtain proprietary data from industry-leading providers, which generally is a pretty hard thing to do if anyone has ever asked for data from CROs. But we're able to use that data and actually do quantitative analyses and actually allow researchers to do quantitative analyses as well. But before I do that, I, of course, have to answer the most pertinent question, at least for us, which is, what is scientist.com? So you can think of scientist.com as a resource for kind of removing any obstacles that stand between you and getting your work done. We work on both sides of the aisle with respect to translational research. So we work with most of the top global pharma, a myriad of biotechs, startups. And on the other side, we have well over 4,000 suppliers in our extended network that we're generally in contact with fairly regularly. Now, next question is, why scientist.com? Why does an industry like this or a company like this exist? Well, if you take a look at this slide, the first thing we have to think about is how much time or where does the most of your time go in research? And unfortunately, the answer is not in experiments and data analysis. It's actually in regulatory processes. So for anyone who's tried to outsource any work with a CRO, you've maybe found it to be a headache unless there's an existing partnership between you and your organization. And of course, the larger your organization is, the harder and more bureaucratic it gets. And so that's kind of, what this looks like. We have an experiment idea. Ideally, we'd like to outsource it, and we're going to do our due diligence of Googling, looking at PubMed and looking at different CROs. Once we've assembled a list, we're going to start contacting them, negotiating, and then once we come across one that we'd really like to work with, if they're not already a vetted partner of your organization, there's going to be several weeks, if not months, of regulatory processes, legal compliance, all that kind of paperwork. So it could be months before you even get work started. And obviously this is a problem. So that's the beauty of scientist.com. It makes outsourcing or just getting or sourcing anything, whether it's services or products, a lot faster so that you don't have to deal with all of the headaches of the red tape and those obstacles. So that ideally, you can actually get work started within a few days. So I know a lot of us, myself included, we tend to like to do things ourselves. There's the thought, if you want it done right, then do it yourself. So that's the quality concern. And then, of course, again, there's also the compliance and the regulatory concern that it's going to take forever to get any work done. Well, luckily, both of those things are taken care of with scientists. So basically, you can think of us as matchmakers for science. We facilitate the connections so that you can get things done a lot faster. And we can do that with respect to many different research areas. Oncology, of course, is a huge space. We see thousands of oncology requests on our platform all the time. And, we are also always expanding our capabilities and trying to add more and more resources that will be useful for researchers. So we do offer, reagents and products, that kind of thing as well. We can get sizable discounts. So that's kind of a newer offering. The Disease Model Finder is another one of those offerings where we're trying to just speed things up. All right. So hopefully I've kind of described why scientist.com is, a resource and why you'd want to even think about using that kind of platform. And if any of you are from any large pharma or biotech, chances are you probably already work through us without knowing it. So whenever we're talking about tumor model sourcing, there's additional challenges. So the compliance one is probably going to be the most familiar, right? If we're talking about human samples or xenografts, we have to think about patient consent forms. That's going to be a thing that compliance or procurement teams care about. Potentially animal welfare, that's going to be another big one. But even besides that, which scientist.com helps take care of, the searching, that's another huge time sink, right? And in cancer, it's way worse because there's so many different mutations that we may be looking for. Potentially, we're looking for, synthetic lethality, or we're looking at metastatic models with resistance to multiple standards of care. And then if we do find models across different CROs and other providers, it's very difficult to compare those models, again, because the data surrounding them is generally proprietary. So that's kind of the reason and backdrop behind why we decided to create the disease model finder, because we can leverage our relationships with suppliers and actually obtain that data that they would otherwise be very hesitant to hand over to just anyone. And we can make it such that you can do quantitative comparisons. You can search for specific gene mutations or differential gene expression, potentially drug response as well, all in a centralized database. And in terms of who is participating, we do have a number of big players that I'm sure are very familiar to most everyone here. But perhaps even more importantly, we have a lot of partnerships with providers that you're maybe less familiar with. And this is another key thing, when you're searching for tumor models, your first thought is to go to the supplier that you're very familiar with, the Crown Bios or the Charles Rivers, right? But what happens when they don't have that model that you need, which inevitably is going to happen once in a while? So you start Googling and turning to these smaller CROs who may not have their own databases. And so it gets even more difficult to find out what's out there. Now, luckily, smaller providers actually love this resource because it gives them extra visibility. They don't have to create their own database, and now researchers can see them. So ultimately, it is a win-win on both sides. So that being said, I just want to go through some stats to give you a sense of what this database looks like and what kind of models that we have there. So here you see a distribution in terms of cancer designation. Perhaps nothing surprising here in terms of where the most models fall. Obviously, we have a lot of colorectal and digestive system cancer models, which you'll tend to find, that'll be the case in other databases as well. Those cancer types which historically are hard to find good models for, like prostate cancer, obviously there's less of those, but we do have a few hundred. In terms of in vivo versus ex vivo, this is always a question. A lot of the times we're just working through cell lines. Maybe we're looking at syngeneic models. We might not want to be spending for the 6 figures for a comprehensive PDX model study. But when you do get there, it'll be a lot easier to search for them. But we do have a lot of listings with respect to cell lines, patient-derived cell cultures, or if we want to get arguably a little more physiological, we can also think about PDX-derived cell cultures and PDX-derived organoids, which some suppliers also will list. So we can dig a little deeper in terms of model types. You'll tend to find that there will be more PDX models than most other things just because there are thousands of PDX models out there. That's just the way it is. But in some cases, such as in prostate or leukemia or in the lymphoma space, we'll find a lot more cell line derived cancer models. So another question we get a lot from researchers when we're giving our trainings is, well, what's the distribution of pediatric or metastatic models? Obviously, these tend to be harder to find. Obviously, we're going to have a lot of adult cancer models, but we do have a few hundred models where suppliers have designated the patient's ages, and so we do know that those are indeed pediatric. In terms of metastatic, we do also have several hundred metastatic models listed as such. And if we zoom in a little more in terms of the breakdown of those metastatic models, we'll find that predominantly we have a lot in the colorectal, skin cancer, breast cancer, and as well as lower and lower as we go down the rankings here. All right. So that's just to give that brief little synopsis of what you'll find in the database. But I think what becomes especially useful is now when we want to make quantitative comparisons that we otherwise wouldn't be able to. So in terms of the data that we ask providers to give us, we do ask for genomic information. So generally that will involve whole exome sequencing. It could be a targeted oncopanel in the case of a provider who's maybe a little more hesitant about just dumping an entire whole exome sequencing data set, though a lot of them do provide their entire set. This gives you an example of the things that we see in the distribution of mutations across KRAS and P53 in our database. So perhaps as expected, we do have a lot of models with constitutively active KRAS. So that's again something you'll see in other databases. We also ask for RNA sequencing data. So we know that in a lot of cases we're looking for upregulated or downregulated gene targets. That's an important thing that we need to search for whenever we're thinking about tumor models. And a very important question that we get from time to time is, well, how do we handle that RNA sequencing data? It is a conventional challenge that when you're dealing with data from multiple sources, in transcriptomics, there's going to be batch artifacts because these providers are using different sequencing platforms. So we do our due diligence of implementing surrogate variable analysis and other methods to account for that variability in batch, and that way we can actually faithfully make quantitative comparisons between the models across these providers. And so what that allows us to do in a way that really just can't be done otherwise is to finally get all these proprietary models and this data all together and conduct different types of analyses. So that for the first time, we can correlate models across suppliers, across model types, and across cancer types. And as you see here, we'll see that models do tend to cluster according to cancer type. And perhaps even more useful is our ability now to use that RNA sequencing data to actually suggest similar models, either from different suppliers or across model types. So what we tend to see on our platform from time to time is a researcher that is looking to outsource a PDX model study. They know it's going to take some time. It may take several weeks, if not a few months, to complete the project if it goes through to completion. And in the meantime, wouldn't you like to perhaps get the most similar cell lines or even patient-derived cells that will allow you to do some quicker tests and just be a lot more efficient in terms of finding the answers to your scientific questions. So in this case, we can take a colon PDX model, and if we know that our drug candidate's MOA implicates a certain pathway, like the Wnt signaling pathway, we can take that gene set and conduct pairwise correlations throughout our database to find the most correlated cell lines or other type of model, depending on what you want to work with. And then we can go ahead and request those and work on them in the meantime. All right, so coming close to a close here. So at the moment, we do have well over 10,000 models listed in this database. So again, that does encompass PDX models, cell line-derived xenografts, or just cell lines themselves, organoids, whether those are patient-derived organoids or cell line-derived organoids, even PDX-derived organoids. And what we can do is, we can use a lot of different filters to really find what we need. Just to kind of point a few out, you can search for your preferred suppliers. That's probably the first thing you'd want to do. If your preferred supplier already has it, what you need, then great, go with them. You can also search for models that have been published in the scientific literature. So we've done, a priori searches before, putting all this data up there so that that these actually do fall in, the scientific literature. You can search for specific genetic mutations and also differential gene expression and even drug response. Additionally, with the RNA sequencing data that we get, we're able to construct a UMAP plot. We'll call this the gene expression landscape, but mainly what it's good for is that quick visual QC. So if you're looking for a very representative colorectal cancer model, for example, this is a great way to do a sanity check and make sure it's like most of the other colorectal cancer models, lest you get a model that just happens to have a very unique mutation that makes things quite different. And so other than that, the goal at scientist.com is really to accelerate science. That's, one of our mottos is faster science. Before scientist.com, things can take months. This is, absolutely the case across the industry. But with all of the universalized agreements and everything that we do to really speed up the regulatory processes in your organizations, we generally can start work even with a new supplier within just a few days. And hopefully now with tools like the Disease Model Finder and more that are upcoming, that will speed things up even faster. And just to prove to you how much we care about your time, I'm going to go ahead and close a little earlier and give you back some time. I know that we're all hungry, so I'm going to go ahead and stop right there. I do invite you to scan the QR code. Just take a look at the Disease Model Finder in your own time and see if it's useful. Please feel free to reach out to me, either with any type of feedback, positive or negative. There are still things we're improving from day to day, and researcher feedback is critical for that. So thanks again, and I'll take any questions anyone has.
Does anyone have any questions? Oh, I'm just bringing I'll give.
So again, nice presentation. In the list of models that you showed, I didn't see syngeneic model.
Oh, did we not include it? We definitely do have syngeneic models. So that is a good question. It could be an oversight.
Or I did not see it.
Okay, we definitely do have syngeneic models.
Okay.
Oh yes, there we go. I don't know if this laser pointer will work, but a little harder to see.
Okay. Thank you.
Mm-hmm, of course. For which one?
If you wouldn't mind.
Metastasis.
Oh, I see. So in terms of, the ones that have been denoted as being metastatic, there's, looks like over 500. So it could be the case that a model might still be metastatic, but that the supplier just does not have that information. That sometimes does happen. That's why we have quite a number that are not specified. So it could be that there are more.
Any other questions, Javier? Okay, no. All right. Thank you so much once more.